thank you for joining us for our next homegrown candidate in the webinar series. I'd like to introduce Cameron Jack today, who's an assistant professor at our department in the Honeybee Research and Extension Lab. Cameron has a 70% teaching, 30% research appointment. The initial, are you from Utah originally?
No, I'm from Nevada.
Nevada, close. So he started as a higher education, a bachelor's from Southern Utah University, then moved to Oregon State for his master's, and then moved from the Northwest to the Southeast and came to UF for his PhD. In 2018, he moved into an instructional faculty role where he was kind of tasked with helping to expand out our beekeeping educational program. And then in 2021, moved into his current role as a tenure track assistant professor. Cameron has been responsible for developing 9 courses in beekeeping. 5 of those are taught regularly, and then also there's some study abroad courses in Thailand that he's engaged with, and also a undergraduate research tour class that he's working on. And sometimes toxicology has been thrown in there. Routinely teaching about more than 500 students a year in our bee courses. So it's really a testament to how much that program has grown. Also, for a period of time, serving as the education coordinator. So for the research side, Cameron is really focused on the importance of honeybee health, and particularly on varroa and some other controlling pests and diseases in honeybee colonies. I'm sure we're going to be hearing about some of that and his teaching today. So Cameron, take it away.
Thank you. All right, thank you. So thanks for the introduction, Andrew. I just want, I've been watching seminars for a while, right? And like before somebody goes up for promotion, they're often giving a seminar. And there's 2 different strategies that I've witnessed, right? You can either tell an interesting story with like just 1 or 2 projects that you take all the way to completion, or you can kind of like try to do everything all at once, which is a little bit more chaotic, but that's kind of the what I've chosen. So I'm like already setting the stage of like, it's going to be a little bit disjointed because I'm trying to jam a lot of things in there, but hopefully you can follow along and tie this thread with some of the work that we're doing related to honeybee health. So as Andrew mentioned, I've been at UF for a while, and in this position as an assistant professor since 2021. But to kind of really like understand like why I'm doing a lot of the things that like my lab has been doing, you kind of have to go way back and like really far back. So we're gonna start in like the early 1980s. This is my grandpa. Right, so this is my grandpa, Gary Cameron, who's a high school principal in Southern Nevada, and he is supplementing his income through bees and beekeeping. So he managed about 150 colonies or so in the hot Mojave Desert, super hot and dry. It's actually not great for beekeeping, but in the summer when the snow is melting in southern Utah, it's a lot of mountain meadows and lots of wildflowers. So great place to make honey. So cool off his bees a little bit, take them up in the mountains and make lots of honey, and bring it back into Southern Nevada and sell honey. So I kind of grew up in just this beekeeping family. I like to tell people, it's like I liked bees before it was cool, because when I was a kid, it was weird. And I remember like people coming to our house and like going into our backyard and seeing bees and like, what's that? It's big rainbow stuff. I'm sorry, you know how it is. And I have this distinct memory of my grandpa. Also, just he was an educator by trade, like so he would come and teach, and I remember him going to like county fairs and going like doing church events and all sorts of stuff. Like he was always talking about bees and beekeeping. And I have this like one distinct memory of me in 2nd grade of my like we're sitting all down and the teacher's like, we got a surprise visitor today, and my grandpa didn't tell me he was coming. He walks in with a card and it's got a sheet on it, and I just remember being like, oh no, please. These are my colleagues, and my friends. Like I can't. Like, this is too embarrassing. I remember he pulled the sheet off and it had an observation hive, and there was, like all these bees, and all the kids were like, whoa. And I was like, yeah, it's my grandpa. It's the first time I was like excited about him. So I didn't think I was going to have like really anything to do in my career related to bees. I just knew when I graduated high school, I knew that I liked science. I didn't really know what to do with that. And so it was kind of a windy road, which is a story that I don't have enough time to get to, but ended up really doing what I truly do enjoy. It ticks all the boxes of science, bees, and teaching, and just world travel. Like it really gives me everything that I really want in a career. And it's cool that I get to follow along with a path that like a lot of my family has chosen. So I still have cousins in beekeeping. I have a connection to the industry. So I wanted to start with that because that's important to understand why I do what I do. A lot of my research is very applied. A lot of my teaching is focused on helping the industry because that's something that's really important to me and I feel very connected to. So while I was doing my PhD, I was here for a couple years and there was a new position that was being created. That was an apiculture lecturer and a distance education coordinator position. And I was 2 years into my PhD and I was asked to serve on this committee, the hiring committee for this. And we had sat down and kind of had our first meeting to talk about like what this position was going to look like. And as we were talking about it, all of a sudden I was getting really excited. I was like, I want this position. This sounds really great. And they're like, well, you're still doing a PhD. I was like, well, if you had a master's, could you do this? They're like, yeah, technically. I was like, then I'm withdrawing from this committee and I will be submitting an application. And so I did, and then I was really fortunate enough to get it. And there were some things about this position that was really interesting to me. I mean, there was a line that's here. I know this is too small for you to see, but what it says is that the primary focus of this position will be to develop a premier apiculture education program. So that word premier to me at the time, I was like, yeah, we're the #1 entomology department in the world. We should have a premier, the highest level beekeeping education program. But what I really came to find out was that word premier is actually kind of profound because premier just also can mean like the first, right? The first thing. And what I had learned as I started getting into this, once I had the job and I was trying to develop this beekeeping program, I was looking for other beekeeping education curriculum, and it didn't really exist, not in the United States anyways. There wasn't a type of curriculum that could train you to enter into the beekeeping industry. If you think about other agricultural commodities, for instance, right? If you're really interested in like cattle production, you could take a bunch of courses that are related to like bovine health and like cattle industry and then do an internship and then go into that industry. If you're really interested in the plants, right? You could take a bunch of horticulture, plant science, plant health courses, do internships, go into the industry. That doesn't exist for bees and beekeeping. And so we were really trying to create a premier beekeeping education program. So that was, I had that kind of tough job at the very beginning because I still had a couple years left to finish my PhD, but I was working to develop this beekeeping education program. And as part of this position too, it was also to be the distance ed coordinator. And at the time that I started in 2018, we had 5 online non-thesis master's students. And we also had about 10 that were in the graduate online certificate program. And they were mostly like medical entomology and a couple landscape pest management, right? So when I got hired, I was given a big stack of pamphlets that explained our online program. And they were like, okay, you go to ESA and you hand it out. And that's our marketing program. So, okay, I'm not going to do that because I don't think that's going to work. And that sounds something I don't want to do. So I looked around at what other people were doing in our within IFAS in the microbiology department had hired a marketing company to do it. So I started asking them information and I started sitting down and met with a marketing firm. And then it took a while, but we kind of got some support in the department to go for it. And so now we have like hundreds. So, and it's been able to be really great. We were able to hire Dr. Georgette Cluiters, who's taken this program and just like elevated it even beyond anything that I could do. So it was really fantastic. And the apiary education or like the apiculture education program has really taken off too. Andrew mentioned that we took 9 different courses. I did teach insect toxicology for a while as well, and took Dr. Bloomquist's course and turned that into an online course. And now Amaya is going to continue to teach that. I don't want to keep beating this too long because I want to actually spend some time talking about the research, but just very quickly to kind of put this perspective, this is like the core unit of the apiculture education program. So there's beekeeping 1, beekeeping 2, and honeybee biology. Probably most of anybody that's been a student in this room has taken beekeeping 1 with me. I mean, everybody takes that class. It's really, I hope they enjoy it. It's a good course, but these are all online, right? And just kind of put this in perspective, just last fall and this spring, we have 700 students in just these 3 classes, right? So, I mean, it's just really growing and kind of getting unwieldy to the point where we needed also to bring in Chase, Dr. Kimmel, the ringer, is what we call him. So he's going to continue to make this elevated even further because we have so many students that are interested in this. Think about it, that's like more than 1% of UF students are taking beekeeping. So that doesn't sound very impressive, but I think it's cool. So a lot of people coming through this university are taking this class. Also, we teach a couple of hands-on courses. So there's the practical beekeeping course, which I've taught 7 times now. This hands-on course where the students get to build the equipment and I give them a package of bees and they have to keep it alive. We do all sorts of hands-on stuff. And then I was one of the first in the part after Christine Miller, when I first got hired, she was the one who really encouraged me to teach a cure class. And there was funding for it at the time for the development. So I've taught this class actually 5 different times now. And had lots of great students, and some of my best memories are with these hands-on classes. And then for fun, because I told you world travel is like something that I needed, we got to do develop a study abroad course with doctors Gotama Swanapong and Sanchai Nari, and Jamie Ellis and I have taught this class a couple times now. Where we get to, there's 2 parts to this class. It's 4 weeks in Thailand, where we get to go trek through the jungles and look for different bee species, and it's a blast. And we have a lot of fun doing it. And then in the second half of the course, we get to it's called Beekeeping in Asia. And we go all around the whole country of Thailand and get to go meet with different beekeepers that are keeping different species of bees. And we have all sorts of wild adventures, meeting people going out into the jungles. We had instances where people get stung and run off into the jungle, and we have to say it's way more dangerous for you to run into that jungle. There's tigers out there. Stay over here and take your stings. So we have all sorts of crazy adventures that are fun. We get to eat all sorts of interesting foods, and we have a blast doing it. And last thing I'm going to say about the teaching program, I promise I'll get into some research. The bee certificate program finally was official in the fall 2023. I generated this image. I knew you're gonna love this. So this is imagine what I figured, like if I don't have a good picture for this, so I asked AI to generate an image of what a beekeeping student would look, an online beekeeping student would look like. So this is pretty much what I imagined actually. It's like somebody in the bee suit right out in their field. I don't know what this is. I think that's supposed to be a hive tool of some kind. But anyways, so the program is growing. We have 36 people in this online program right now, and people are graduating, and it's growing. Chase is now effectively managing that, and will no doubt continue to take it to new heights. Okay, all right, I had to get through that section first because I wanted to spend a bit of time actually telling you about some of the research that we're doing. And I already told you from the very beginning, my interest as a researcher really comes around honeybee health. Like I have like a unique position where my main interest is in the welfare and like the well-being of one insect, and then also the like industry that's kind of surrounds that, right? And so if we had a bigger space, I could probably come up with like 12 more factors that are influencing honeybee health. I just was trying to list a few of the things that we touch on specifically in our lab. And for the sake of time, I'm just going to narrow this down into the pests and pesticides things. If you look online at like our departmental page and you see it says Cambridge Act, it says honeybee toxicologist, which is a bit of a mystery, if I'm being honest, because I don't claim to be in the lofty realm of a toxicologist. When we have folks like Daniel Swale and Ted Burgess and Amaya, like they're like real toxicologists, and I just kind of stayed in a Holiday Inn Express. I've just done just a little bit of toxicology in my work, but I'm trying, just for the sake of time, I'm kind of going to tell this story with some of the work that we're doing related to pest and pathogen management and pesticide impacts on honeybees. So what I want to tell you about is these 2 species real quick, and we're in an entomology department. I know that you guys know a lot about this, so I'm not going to give time on a lot of background, but we've got public enemy number 1, which is Varroa destructor. This parasitic mite feeds on the hemolymph and the fat bodies of honeybees, and really the issue related around Varroa is that they vector and transmit viruses that make bees sick. And they're really hard to control, because if you use chemical controls, the mites have developed resistance to many of the synthetic chemical controls that beekeepers have used. And so it's really difficult. There are some cultural and mechanical controls, but it's really time consuming. And for a commercial beekeeping industry, they're really just dependent right now upon chemical control. There's not a lot of options. So we'll talk a little bit about that, but I also introduced you to public enemy number 2. That is the small hive beetle, Athena tuta. And this beetle, what it does, it gets in, invades inside honeybee hives. There's a yeast associated with their bodies that will cause the honey to ferment and to kind of get slimy. They lay their eggs kind of like in a biblical plague proportion, like those eggs will hatch and the larva will start feeding on everything and really can drive a weak colony out very, very quickly. So this is, especially in the southeastern United States, this is really, a lot of beekeepers still really struggle with small hive beetles, us included. I mean, it is tricky to manage. So some of this work is going to be primarily focused on controlling those 2 pests. And I want to start off with sharing with this one research project where we have explored a lot related to this chemical oxalic acid. It's an organic compound. It is really kind of a weak acid. You can find oxalic acid in a lot of the foods that we eat, but just like anything, the dose makes the poison. And when you concentrate it, it's strong enough that it's going to kill some mites, and it can also be harmful for us if we're not being careful. So we do have to be careful as we're handling oxalic acid. But this is something that beekeepers have been using illegally in the United States for actually a long time. But it wasn't until about a decade ago, 2015, is when the EPA made an emergency use permit for us to use oxalic acid. The language related to the usage was quite vague and very unhelpful, and beekeepers get creative and come up with all sorts of things when you are very vague. And so a traditional way to apply it is by mixing it into a sugar syrup that you then trickle onto the bodies of the bees. And one of the reasons people really like this is because even though it's been used for decades kind of illegally in the US and around the world, a lot of other countries have used oxalic acid, there are still no reports of mite resistance. Probably the reason there is no reports of mite resistance is because oxalic acid is also incredibly water soluble. So it breaks down very rapidly inside of the hive. So it's really kind of more of a flash treatment. It comes in, can knock some mites down, but then it dissipates really quickly, dissolves, and then it's not around anymore. Now, one way that has been to apply oxalic acid that's becoming more popular and has been for about the last decade is to essentially heat the oxalic acid and to where it vaporizes and you're essentially fumigating inside of the hive. And so the oxalic acid settles all over, it spreads out the bees, it kind of elicits a grooming response. So the bees are going to be grooming a little bit more, you're going to get a better knockdown of mites. Now, I told you that language when that emergency use permit was registered for oxalic acid, the language was very vague, but what it said was you are only allowed 1 gram of OA per brood chamber. I'm not going to get into a lot of this just as background, but during my PhD, this was something that I was interested in studying. We found that 1 gram wasn't nearly enough. It was not effective. And so we had found that up to about 4 grams is really going to be a much better option for you, even though that's was not available to beekeepers for a long time. So beekeepers were asking me, especially in the last few years after some of this work is coming out, they're asking us well, we really want to use OA. We don't feel like we're getting very good control with the other available treatments. So we want to use OA, but which application method is better? There's the traditional method of dribbling. There's also this is vaporizer. This is a different model of a vaporizer. But I told you beekeepers are creative and they're coming up with all sorts of their own things that they're asking me about. So some beekeepers are at home cooking up oxalic acid and glycerin on their stovetop and soaking it into shop towels and putting it in. It's very not a good idea. But people are looking for cheap, effective things, right? And so that's one method that people have been trying for a while. Another is to use insect foggers. This is often used in mosquito control. It creates a nice big plume. And so beekeepers were mixing grain alcohol and oxalic acid and sticking it on this propane torch, which is also not a very good idea. But you can find lots of videos on this on YouTube. And so people are asking me all sorts of these questions, what should we use? What's the best? And so we wanted to try this out a little bit. And so I hired the bravest undergrads I could find to go. I'm just kidding. Actually, that's not true. I did use some undergrads. But they had to sign some waivers. Also, there was, so if you looked at the label, a few years ago, the temporary use permit ran out. And so then a company was able to start providing pure oxalic acid, and it's called apobiol. But even still, the labels were still super generic and saying stuff like it may damage your brood. You should be treating, applying this multiple days apart or several days apart. But then I was getting tons of questions right when this was released. It was like, what does several mean? When should I be applying this? Right? And because several can mean anything. So this was really our crux of our research project. This is one of the first ones I did while I was an assistant professor. Was we wanted to determine which of these methods is going to be the most effective, and essentially at what rate should we be applying this, and how frequently, and what's that going to do to the health of our well-being of our bees. So one of the first technicians I hired was a guy named Cody Crowdy. He was really fantastic. He's now he went to the University of Minnesota, and was doing some great work there as well. So the way we set up this experiment is we had 40 colonies. So we had 10 colonies in each group, and we tested 3 of the 4 of those treatments that I'm just showing you. This is the OA vapor at that 4-gram limit, which is what we had found through previous research. We did the current legal limit for the OA dribble, and then we tried the OA fog. And I had to talk to lots of beekeepers. I watched YouTube videos, took good notes of like what people are doing, and kind of took the average of what everybody's doing with that. The OA shop towel method that I was showing you was part of a different experiment that I don't have time to explain, but it didn't work very well. That's just, we'll talk about that another time. So what we were doing was we were applying this once a week for 3 weeks, just kind of stay standardized. And we were taking kind of pre-treatment colony strength evaluations, estimating the amount of brood, the amount of bees that were kind of at the beginning and post of the experiment. And then we were collecting alcohol washes, so we were collecting a bunch of mites in alcohol, or a bunch of honeybees in alcohol, shaking them, dislodging the mites, and then we can count. And that helps us get some idea of like a mite infestation level. So it kind of lets us know where we're at mite population wise inside of these colonies. We were doing that before we took any of these samples. And so what I really want, there's of course a lot of data to share, and I'm just going to boil this down to kind of the most simple aspects of this, just for the sake of time. But what I really want you to look at here is kind of the way we're going to measure this for now is like our pre-treatment levels and our post-treatment levels. And so you can really just kind of look at the slopes of the lines to kind of get an idea of how things are working. So if we look at this first figure on the left here, this is our number of bees. So our bee population, how it changed over this like 1 month period. And the only group where it significantly improved was this vaporizer group, when we vaporized with 4 grams of OA. If we look at the brood in this figure, there's 2 groups that had like a significantly increase, and that was our OA dribble, which was the legal method at the time, and then the OA vaporizer group at 4 grams. Both of those significantly increased. So we had more bees, more brood. The colonies were looking good. And then kind of most importantly, this mite infestation level. So what are our varroa populations doing in these different groups? And the slope of the line will tell you everything, right? This blue line here is our control group, and control groups do what control groups do. Like if you're not treating for varroa, those mite populations continue to grow. But interestingly, this OA like fogger was not doing anything. It really was not reducing our mites at all. Even though there's like a big impressive plume of vapor that's coming out, it's not delivering enough oxalic acid and not delivering it in an effective way to affect those mites. So the OA dribble and the OA vapor were what was really successful at bringing those mite populations down. So then we went on to phase 2. We felt pretty good about the OA vapor. We wanted to continue to provide some more evidence that the OA at 4 grams was going to be safe for bees, but also be effective tool for beekeepers in hopes of a label change. And so what we wanted to answer was that the second part of our question is like, how often should we be applying this, right? And so we had our kind of repeated a similar experiment where we had 40 colonies. And this time we basically were just doing it at different intervals, always 4 grams of OA vapor at a time, but at different intervals. So 3 days, every 5 days, and every 7 days. And this time, we were also collecting alcohol washes, kind of pre-treatment, post-treatment, so we could keep an eye on our mite populations to see how they changed over this like 1 month period. And then we also were measuring mite fall. So we're sticking sticky boards underneath. And when I told you, when you apply oxalic acid, especially in a vapor, it elicits a grooming response, right? And if we're killing mites, we're expecting them to fall down to the bottom. So we can kind of capture those mites that are falling over like a 72-hour period and get an idea of how well and how effectively are we killing mites. So I know this is a little messy, but just hang with me for a second. So let's look at this figure on the left. This is our 72-hour mite fall. Notice that this blue line here is our control group when we're not applying any kind of oxalic acid treatment. So those mite drop numbers are kind of staying steady like you would expect. And then our first application of OA, they all increased. So I mean, they're all getting a 4-gram dose, right? So they're killing some mites. But it's really what's interesting happens by the second and the third application. When we waited only 3 days to reapply our next treatment, then you see that we really didn't kill very many mites. That's this green line right here, is this 3-day application period. But when we waited about 7 days, we were killing more mites. The same pattern was true by the third application. When we waited more time, we were killing more mites. And by the fourth time, we had killed so many mites that it's kind of just essentially it's about the same. And again, this is probably looking at just a mite infestation through those alcohol washes probably tells the best story of just looking at these slopes. Our control population continued to grow while our 3-day intervals, this green line, you see how it does decrease, but it's only slightly and it wasn't significantly different from our control group. But when we waited every 5 days or every 7 days, we really significantly dropped. That may not sound like super exciting, like groundbreaking research, right? Like this is very applied, but this is something that really has, I think, been a benefit to beekeepers, because there were multiple beekeepers that were telling me that they are so anxious about controlling varroa and are struggling so mightily to control this mite that they have been using oxalic acid. Like there was one beekeeper who told me every single day, which is like, whoa, that's not a good idea, right? But then other people that were saying at least like 2 or 3, like every 2 or 3 days, right? They were really just throwing so much of this, and that's expensive and not effective, right? You really do need the time to allow those mites to come out and emerge, and giving them a little bit of period of time before you can reapply is really important. So I've kind of gotten in the habit, as I share my research with beekeepers, to provide a QR code so that they can scan it, and a lot of beekeepers want to read everything. So kind of as I've gone through, I'll just provide that if you're interested in reading on this a little bit more. But this was one piece of work that I think was helpful for beekeepers, and directly, and it did lead to a label change. The oxalic acid label is now updated, so they updated it, increased it to that 4 grams, and there's specific language now that says you need to wait every 5 to 7 days. So I counted that as a little win of, yeah, I did something. Take that, Grandpa. Not really take that. You just, all right. Okay. So another thing that we were interested in was this a separate class of chemicals, because I told you beekeepers are desperate for varroa control, and there are so few options available for them, especially any synthetic miticides. There's really only one right now that is being used with any sense of regularity, and we're already seeing lots of signs of resistance to that. So beekeepers are desperate and asking us, like, what's next? What's next? What's next? And I had a beekeeper that came up to me a few years ago after a meeting and said, "Hey, I'm a veterinarian, and in our practice, we're often using this class of chemicals, isoxazolines, that are used for veterinary medicine to control fleas and ticks and dogs and cats and stuff. And has anybody looked to see if that would be toxic at all to varroa?" And I searched through the literature and I couldn't find anything. And so my master's student at the time, Julia Saint Amant, decided she was going to take this project and kind of work towards it. And she did a couple of things that I think are noteworthy that I want to mention here. So it's pretty, honeybees are big. They're easy to apply chemicals directly to the body of the bee. And so that allows us to know the exact dose that a honeybee is getting, because I can just put that amount right onto a bee. But really, we have a hard time doing that with Varroa. They are very small, right? And so it's difficult to topically apply a pesticide to a mite. And so at the time, a few years ago, we had read a paper of somebody else in Canada doing this, but it was the only other instance that I had ever seen of anybody topically applying Varroa in this way that would make it kind of more of an apples to apples comparison of toxicity to a bee and a mite. Because previously, you'd always just put mites into a vial that had been treated, but that kind of just gives you an idea of like a lethal concentration that the mites have been exposed to, but you don't know exactly the amount that they received, right? You know, the actual dose. So we took a micro injector that's used for injecting pupa. We found a way to stick it up on its end. And then we could kind of slide these mites around and deliver with a very small needle topical application. It took a while to kind of just play with it, but it ended up working out really well. And that was something that we were able to continue to do to kind of, again, allow for like more apples to apples comparisons. So we can have an LD50 for Varroa and an LD50 for honeybees. So I'm kind of, again, I'm taking a whole lot of data and I'm just trying to simplify it for a quick story so that you kind of understand what we were doing. But what we're looking at now, since we have an LD50 here and an LD50 for bees, we can kind of really answer this question: is it more toxic to Varroa or is it more toxic to bees, in a way that's a better comparison? So Amitraz is a formaldehyde insecticide. This is something that has been used to control cattle ticks for a long time, and beekeepers have been using it actually for a while. We've gotten away with it for a long time as one of like the primary things that beekeepers were using, but we're starting to get our comeuppance now. There's lots of reports of Amitraz resistance. By the time that we were doing this, it was still, as you can see, quite effective. So that, I mean, this was 2,000 times more toxic to the mite than it was to the honeybee. Coumaphos is one of the original chemicals that people were using to control Varroa in the United States, and this is an organophosphate. It's kind of nasty, not a really great chemical anyways, but it was quite toxic. At this point, the mites are so resistant that it's 26 times more toxic to the honeybee than it is to the mite. Right, so it's just not a good option. Some of these that we tested, these isoxazolines, like afoxolaner, lotilaner, ceralaner, they were all like kind of meh, like really not toxic, not worth it to pursue. But fluorolaner showed some promise, right? We saw that it was like 90 is still a far cry from 2000, right? So it's not nearly as toxic, but there's something there. There's something to work with at that level, that maybe with some good formulation chemistry that we can find a way to apply this in a way and that maybe at least is worth exploring. And so I don't have a lot of time to go with it, but we did have a really great collaboration with Dr. Daniel Swale and his lab and some of his postdocs, and Song Yoon, who's a postdoc in Daniel's lab, was doing some really cool stuff. I mean, he was dissecting the central nervous system of Varroa and sticky probes in there and seeing if fluorolaner was going to like actually like cause like the firing of the hyperexcitation of those nerves, and it did, which was really cool data. So this is something that we are planning on exploring a little bit more. So now my new master student, Pierce Barron, is going to continue to do a little bit of work with fluorolaner and maybe some of the other less commonly used isoxazolines. And so we'll continue to kind of explore this class a bit more. So kind of moving on to our control of small hive beetles, right? This is, again, public enemy number 2. And if you think of it in terms of chemical control for small hive beetles, there's really not a lot for us to go off of, right? Right now, there is a ground drench treatment, because I didn't really talk about the biology that much, but if basically when small hive beetle larvae start hatching, they're starting to eat everything in the hive, and when it's time for them to pupate, they crawl out of the hive, burrow into the soil, and pupate in the soil. So you can treat the soil with permethrin, a pyrethroid, to kill some of them, but at that point, the damage is already done. right? Like your colony's already been slimed out and it's been a big mess. So we want to find something that is going to be more useful inside the hive to prevent the damage from even taking place in the first place. The only in-hive chemical that is allowed to use is this product called Checkmate. It's also that one I was telling you, the active ingredient is IFAS, and it is registered for use against small hive beetles. And I told you, I've mentioned this several times already, beekeepers are creative at solving their own problems, and so they're coming up with things that are quite dangerous in many cases. So this is a good example. To kill small hive beetles, they've been using roach baits that they are putting inside a piece of corrugated cardboard. The idea is that the beetles can kind of get through, and as they're hiding from bees, they can kind of go and hang out and hide in there, contact and eat maybe a little bit of this roach bait, which is traditionally the one that most beekeepers have been using. Is the active ingredient of that is fipronil, which is incredibly toxic to basically everything, especially honeybees. And so it's not like a really great safe idea, but that's what a lot of beekeepers had been doing. So Julie and I had been talking about this, and we kind of thought, beekeepers are going to do what beekeepers are going to do, and we don't have a good effective treatment. So what if we could at least, we know that fipronil is incredibly toxic to honeybees. What if we took a look at some of these other roach baits that are out on the market to just see what's out there, right? See how toxic they really are. So some of the ones that we tested with these other brands, that had the active ingredient of indoxacarb, abamectin, hydromethanol, chlorothianidin, fipronil, and then this was our industry standard of that Kumafos. And so, for time, I'm just kind of, again, taking a lot of information and kind of shoving it into a single slide here. And this is our topical LD50 that we're seeing. And basically, the results were really disappointing. Essentially, everything was very toxic to honeybees. These, like abamectin, was 813 times more toxic to bees than it was to small hive beetles. So it's really not a good option. But everything was really bad. Even the fipronil, what beekeepers are primarily using, is essentially the same, like 1-to-1 of toxicity from bees to beetles. So really not a good option. So the takeaway that we are doing now is just kind of trying to get this information out there. Like, hey guys, it's not worth it. We'll get several calls a year of beekeepers that are finding like a bunch of dead colonies, and when you go into that, you find that they have those like homemade beetle traps where they're putting in the fipronil, and if it gets a little bit of water in there, that's going to leak out, and it's just going to decimate the hive very quickly. So it's kind of disappointing news. We're like, hey, we don't have really a good solution for you, but we know that you shouldn't do what you're doing right now. And that's kind of a lame story to tell. So we wanted to at least explore a little bit more. This was a project that actually started with some undergraduate researchers that Julia was kind of supervising. But we thought, we know that there are other beetle pests that we have in the United States that are also being controlled with different pesticides. We were kind of doing a quick literature search and finding out some of those, what chemical groups that are being tested for them and being used to control those other beetles, and looking to see what we already kind of knew in terms of honeybee safety, to see if we could screen any of them. So this is kind of a weird hodgepodge list, but this is what we came up with. Neonicotinoid, that's acetamiprid. Then we have chlorotrimilprol, flubendiamide, propiconazole, which I know is a fungicide, Daniel, but people use it for controlling larvae in carpet beetles, and then we also have thiacloprid. So kind of a hodgepodge groups of different chemicals that are different insecticides and one fungicide that people have been using to control beetles. Again, lots of data that I'm kind of jamming together, but kind of the short and sweet of it was that some of these were really not going to work topically. Some of them worked a little bit orally, but really some of them really will only work against small hive beetle larvae, which again is really not my goal. I feel like we can kill larvae, but by that point that there's beetle larvae in the hive, the damage is done. I want something that's going to kill the adults. The only thing that was really exciting from this was acetamiprid. Now, acetamiprid is something that definitely has some potential. We are not necessarily the first ones to look at this. Some Italian researchers, including Ilaria, has done some work with acetamiprid in the past, and so this is something that we want to kind of develop further, and we're doing that right now, trying to find ways to safely apply acetamiprid and look at how that might impact our honeybee colonies. So this is kind of a bit of background, about some of this work that we're doing with beetle or honeybee pests. But some of the, I think successes that have come from this research is that we have been able to provide some information that's led to a label change for the use of oxalic acid. We've found maybe a new avenue of something to at least explore with fluorolaner. We've hopefully are able to more effectively warn beekeepers about the dangers of using roach baits inside their hives. And we're maybe exploring more with acetamiprid in control of small hive beetles. All right, so I've got a few minutes left. The last kind of story I want to tell is about some of the work that we've been doing with some mosquito control districts related to the impacts of mosquito control on honeybee health. And this is something that I've been hearing, basically all my life is that oh yeah, mosquito control is so bad for bees, right? I mean, you don't want to put your bees anywhere near where they're going to be spraying. And then I moved to Florida, and I mean, we rely so much on the mosquito control industry. Like, we need it. My wife would not live here if there was not effective mosquito control. And so insecticides really are necessary part of this. And so there's always a lot of finger pointing. I told you, I mean, I could come up with a huge list of all the factors that are influencing honeybee health. And when bees die and somebody is spraying anything in the area, people are going to start pointing fingers on what killed their bees, right? Whether it's founded on truth or not. So this is something that we were interested in exploring a little bit. We had FDACs came to us and really wanted to do more of this work. And so we decided, we had a lot of research in this part, but I don't quite have time to get into all of it. So I'm going to break it down into two parts where we wanted to detect the amount of mosquito control products that were ending up physically inside the hives, and then kind of seeing like how much of an impact that had on our honeybee health. And so we collaborated with the Anastasia Mosquito Control District, who, by the way, were fantastic collaborators, just great people, and I loved every minute working with them. They were fantastic, and they were very open with what they were using. They were using mosquito mist, which is primarily the active ingredient is chlorpyrifos, which is used a lot in truck-mounted spraying along roadsides. They have aerial spraying that dibrome, which is primarily nalen, and they had a lot of other truck-mounted spraying of duet, which is these two pyrethroids. Pyrrolithrin is a really potent knockdown agent, and then sumatriptan is enough that's going to kill a lot of mosquitoes. They were also doing a lot of water treatments in the area with this BTI. And so that's even, you might not think about, like, honeybees still collect water, right? They are foraging for water, not to drink, but to bring back into the hive to help with cooling. And so beekeepers, once they found out, oh, they're treating the water, like, that's probably why my bees died. So, we wanted to kind of explore all these different avenues. And so Devin Garan, who's a fantastic beekeeper and field technician, was doing a lot of this field work. And then my postdoc, Lena Barasco, is now since taken a job at the University of Auburn. They were the ones leading this research project. And the whole goal with this is we wanted to stick these colonies into hotspot treatment zones where we knew that they were just going to get hammered with mosquito control. And some of these other areas that were kind of out of the way that, or in no spray zones that would be relatively pesticide free in terms of mosquito control. And we had we had a total of these 30 nucleus colonies. Nukes, we call them nukes for short. That's just is a smaller hive, about half the size of a normal hive, but they're still a functioning colony. And we had about 5 colonies in these 6 different sites. And we had these sites kind of planned out a bit ahead of time with some of the people at Anastasia Mosquito Control. But those folks are so great, and there was a person there that was like, It's like, do you need to do some field scouting of this? I was like, yes, I do. Let's get up in that chopper and take a quick peek of what's going on. So that was probably one of the highlights of the best perks I've had with my job so far. It's a quick helicopter ride around St. Augustine. So these were the sites that we had chosen, and we chose this based on history of what Anastasia mosquito control was. They knew that we're going to need and require lots of treatment. And then we're also starting to collect samples. So we started this experiment in August, mid-August of 2023. And just prior to that, we had collected samples from all of these nucleus colonies, collecting honey, wax, pollen, larvae, and we were collecting water nearby before we started. And then we ran this for about 3 months. So in November, we collected these samples again and did these residue analyses. And so this was really actually good timing for this research. If you remember, in August of 2023 is when we had a Hurricane Idalia come through. It was a big Category 4 storm that tore up Port Cedar Key and then went across and just dumped tons of water in kind of like North Florida. And so while we were doing this experiment was when they were treating like crazy around these colonies. And so experimental wise, it worked out for us to time it in that way. So what I'm going to show you here is just a couple figures of the percentage of pesticide, of the samples that contained pesticides. And we have here like kind of pre-treatment and post-treatment here. So you kind of notice some things right away, right? With the pollen anyways, this was the only one that was like a significant increase from pre-treatment, like pre-spraying to post-spraying. We had a significant uptick. We had a lot more pollen that was showing more with pesticides present. But you can see with the just like pre-treatment, anyways, we had like 20% of our samples still showed up with a lot of these mosquito control products, even before we were doing any kind of mosquito control, right? So some of these chemicals are just in the wax and kind of present. And when we look at which of those chemicals were showing up in wax and the pollen, primarily it was chlorpyrifos. And chlorpyrifos that's what's used in the truck mounted spraying. This is also a chemical that's used a fair bit in agriculture too. So it's very possible that we were picking up so much of this essentially from agricultural areas. And we didn't find any nalid, but we found nalid's breakdown product, dichlorobos, which was detected in some of the bee samples, and we found a fair bit of sumethrin as well. So kind of keeping this quick and simple of kind of saying what we found. I kind of put this in a table that I'll try to just break down quickly for us. So up here we have our untreated sites, and down here we have our treated sites. Here is kind of before treatment, and here is after treatment. So when we're looking specifically at chlorpyrifos, which we found was the only chemical that we found detected in pollen samples, we found actually a fair bit in both our untreated and our treated sites. But we actually had the highest hit, 71 parts per billion was found in our untreated site, right? Like afterwards. So that is most likely probably doesn't have anything to do with the mosquito control. Probably has something else. It could be mosquito control, maybe from some drift, but most likely it probably has something more to do with some of the agricultural areas that they're in. If we look at dichlorobos, which again is that breakdown product of nalid, we only found it in our treated sites, and we only found showing up after mosquito control treatment. But it's still at a really kind of small percentage. I mean, we found it in 10% of our honey samples and only 3% of our wax. But even still, it was at incredibly small amounts. 0.8 parts per billion is not much to be worried about there. And with sumethrin, this is that other product that's part of the duet treatment. We only found it in our untreated sites, which again, this is probably being used in other ways that's not by mosquito control. So we're really just, to answer that first part of the question, we're not seeing a ton of these chemical residues showing up inside of the hive. But we also are trying to, we also of course want to see what does that do to colony health. Well, so we were measuring like the amount of bees and brood, and kind of really anticlimatically, like didn't do anything, right? Like, I mean, this is the number of bees per colony. You can see that our treated and untreated groups were essentially the same over this 3-month period. And that is also true for the brood. You can see that both the treated and untreated groups declined in brood over the time, but that's really probably more of an effect of the season. I mean, even though it's warm in Florida and we don't usually go completely broodless like they would in other really cold climates, the queen is still slowing down her laying, and so they're still shrinking in size. And so, but it's not shrinking at any different rate. So this was actually, I think this was great news. It was like best case scenario for me. I could go to the beekeepers and like, hey, chill out. Like, it doesn't seem like this is a major issue right now. But then I also, the mosquito control districts are all high-fiving each other. Yeah, like it's not, but this is just, of course, one specific instance in our area with one particular mosquito control district. So certainly there is, potential room for things to go sideways and go wrong with mosquito control in honeybees. We have, there's more work that's been done that Lena is now publishing, where she was taking all those same active ingredients, testing them at those same concentrations that we were finding in the field, finding that they really weren't too impactful, but if you increase those to some degree, then it started to really pose some problems. So as long as it seems that as mosquito control districts are applying some of these treatments responsibly, the impact really should be minimal, if anything. So with this, I think it's been helpful for us to continue these conversations with beekeepers and mosquito control districts and try to calm down some of the tensions that that people have been experiencing. It's never going, I mean, we're still a university and some beekeepers are going to be like, I don't trust anything that you say. It's not going to calm everybody down, but hopefully this is going to kind of diffuse some of the tension that as we kind of get this message out. So that's where I want to end it for now. I just wanted to specifically just tell you guys, like, we have just really the best honeybee lab. And you might be thinking of the building. The building is cool, but the building is just a building. What makes the honeybee lab so awesome is because we have great people. And so we just have an excellent team of researchers that are super dedicated and work hard. And so if there's been any kind of success that we've had, it's really because we just have an awesome team assembled. So anyways, I'll stop talking for a second. Maybe we got a couple minutes for questions.
Some questions.
A couple questions.
Yeah. At one time, they were selecting honeybees with increased grooming behavior to control the varroa. And I wonder if the oxalic acid vaporization is working because it's stimulating grooming behavior.
Yeah, so I think that is certainly part of it. So if you didn't hear the question, there's some research where people have been selecting for these traits of increased grooming behavior. Because remember, I didn't have time to get into all this, but varroa is not native to Apis mellifera honeybees. So we have to kind of select for these traits of bees that are better hyper groomers, right? And so oxalic acid certainly is eliciting a grooming response. However, I can still take a mite, I can hold it down, and I can put a little bit of oxalic acid on it, and I can see its heart rate increase, and it will die. So there's something still molecular that is happening, and we actually don't have a really good handle on that yet as to like the mode of action of oxalic acid. But certainly, I know Daniel, I saw it. We need him on this. But certainly, I think this is a component of part of that is the grooming response. What else? Yeah.
So I'm curious if there's no detectable effect of chlorpyrifos on the honeybee health, but are there, as you may have seen, people are freaking out from finding glyphosate in bread at certain levels. Are there any implications of finding chlorpyrifos kind of ubiquitously in pollen or other honeybee products? So
there's always, essentially, anytime we look at pollen, we find stuff. and every time we look at wax, we find stuff. Like honeybees are interesting. I mean, bees in general, like they're touching everything and bringing it back, right? And so there's always going to be some of that. So certainly there is. This is why pesticides will never really go away with some of the blame. Like it's so easy to point to that as an impact. And certainly, I mean, pesticides have an impact. Honeybees are insects, but to the level, we're not usually seeing that, at least here. So I guess I haven't heard people really chattering a lot about chlorpyrifos yet. But I suppose it's there for those that are really focused on that. Yes, sir, Norm.
Last time I was involved in this issue, one of the efforts was to go to hives where the bees are dead and sample. And I think the state did quite a bit of that. Do you know the outcome? Did they find things? Did they not find things?
Yeah, this is interesting, and this has been actually in the news a fair bit, because so when there may be a couple different examples here that you might be referencing on, like a few years ago, actually 2 decades ago is when Florida was like all over the news. When this is when like the term like CCD like was really coined, was this colony collapse disorder where bees were just missing and they were trying to sample things, and basically what they determined was it's a mystery. So that's why there's potentially multiple effects that are causing bees to just kind of straight up leave. But then there in the more recent history, like there have been like massive die-offs in the last year or so affecting commercial beekeepers. And the research is somewhat beginning and like a little bit confusing, because they will certainly find some levels of pesticide, but it's not particularly high. They're finding deformed wing virus, like a virus that's vectored and transmitted by Varroa, but it's not always really high. But they're finding one virus, acute bee paralysis virus, that is a little higher than normal. It seems to be quite toxic. So a lot of people are pointing or deadly, but a lot of people are pointing the finger at that, but it's probably not just that. It's never going to be like one thing that's like a simple, quick fix, right? So there's still a lot of confusion and a lot of I think that's why it kind of helps spawn, at least with beekeepers, a lot of anxiety, is because we have a really hard time pinning down some of these root causes.